Hi, everybody. Welcome back from the lunch break. I'm very excited to introduce our speaker for this afternoon. This is Dr. Dina Katabi. She is our keynote speaker. She is also the Thuan and Nicole Pham Professor of Electrical Engineering and Computer Science at MIT. She's a director of MIT Center for Wireless Networks and Mobile Computing. She's a member of the National Academy of Engineering, a recipient of the MacArthur Genius Award, and she has many, many other accolades to her name. Dr. Katabi is a fantastic innovator, and she's going to speak to us today about a really exciting and cutting-edge topic in machine learning, wireless sensing, and health. I want to remind those of us who are maybe joining via the live stream that if you have questions for Dr. Katabi, please enter them into the chat, and we'll get to you at the end of her talk. With no further ado, please welcome Dina.
So let me start by thanking the organizer of this conference for two things. Of course, I want to thank you, Zach, Mazier, everyone here first for inviting me. But more importantly, I want to thank you for creating a community. Like, look at the people in the audience. I mean, you have medical doctors, you have computer scientists, you have health professionals. Those people typically we don't have a community, but we have this growing field of AI in medicine, AI in health, and creating a community is essential, so thank you very much for working on that. And let me start my talk. Okay, can we get the slides? Sorry, I like to be close to the people. So it would like be threatening if somebody is going to ask a hard question. You know, it's like look them in the eyes. Okay. So what I'm going to talk about today is measuring the brain, its diseases, and therapies using AI. And this is a topic that is very close to my heart. Of course, this is something that my lab at MIT is working on. And one natural question that I get from people typically is why to use AI to measure the brain? Like, why the brain in particular also? So one of the things that is really important is because we really don't have really good measures of the brain. So when you are talking about diseases like neurodegenerative diseases like Parkinson, Alzheimer's, Huntington, you name it, you ask questions to people. You ask, you don't have like a good, like, we have some imaging, but it is expensive, also it's not conclusive. And we just rely on questionnaires, PROs. Similarly, when you are talking about mood disorders, whether that is depression, anxiety, etc., bipolar, it's really questions. And can we really have measures? Like if AI can help us, that would be great. The next question is how? How would AI help us in measuring the brain and its diseases? Now, of course, what I want from AI, I want a measure that is easily accessible, a measure that anyone can use anytime, because that would allow us to reach to all people, whether they are next to the clinical center or they are in a rural area, from all types of, like whether they can answer a question, whether they can measure themselves. I want something that is the easiest possible. So what is easier than just having to do nothing, really? So basically, imagine with me if we can have something that looks very much like your Wi-Fi box at home. So it's a wireless device that sits in the background of the home and uses AI to analyze the radio signal that bounce around, that bounce off the human body. And from that analysis with neural networks, be able to get your sleep, know when you enter REM versus when you enter light sleep or deep sleep, get your breathing, your heartbeat, get motor symptoms if we are interested in diseases like MS and Parkinson's, get all of that stuff, get the slides on the screen, actually. What happened to the slides? I see. Okay, good. Okay, So basically, can we do that with a wireless device? And the way we do it is, as I said, using AI to analyze these radio signals that bounce off people's bodies without asking people to do any measurements themselves, completely passively. We have developed this device in my lab at MIT. We call it the Emerald Box. And I want to show you some of the result that we get, and then move to actual brain diseases and show you what we can do there. So this is one of our early results. So this is one of the offices that we have at MIT, and this is one of my students. We are monitoring this person from the adjacent office. So you see a big arrow. So the device is not even in this office with this person. It's going to monitor him through the wall because wireless signal traverse walls. Okay? So let me play this video. And look at this red dot at the side of the screen, the small red dot there. So this is where the device thinks this person is walking right now. And as he walks, you can see that the device knows as he's moving without any camera, without seeing him, just through the wall, actually, without him wearing any sensors on his body. Now, of course, everything that we do, we publish, so it is peer-reviewed and compared with the gold standard. In the case of motion, we use something called akin motion capture. Like if you went to Spalding, for example, in Boston, rehabilitation hospital, they have these big rooms where the ceiling is planted with infrared cameras. You put infrared markers on people, and that's how you measure their motion. We compare to that, and we show very high accuracy. And also, we can monitor multiple people. But in this graph, in this result here, I showed you a dot on the floor map, and I told you this was one of our earlier results. So let me show you one of more recent results. So here you see the big frame actually is what the device sees, what the wireless sensor can capture. So now it can capture a full skeletal representation, a stick figure of the person. And this scene at the top is just what's happening inside the room so that you can understand whether the wireless signal is getting the stick figures correctly. So now if I play this video, you see when he sits, actually you see the stick figure sitting. When he stands, you see the stick figure standing. When he moves his right foot, you see the right foot. When he moves the left foot. And again, remember all of this without any wearable devices on the person. Nothing. It's just because when we move, we change the electromagnetic waves around us. And with neural network, we can capture those changes even through a wall. We can also monitor sleep. So when people go to sleep, our brain waves change and we enter different stages: awake, light sleep, deep sleep, and REM, rapid eye movement. Sleep stages, of course, are very important for sleep disorders. In the US, 1 in every 3 people have sleep problems. But actually, sleep is a platform for almost all diseases, and particularly neurological diseases. So just to give you an example, REM tends to happen early with people who have depression early in sleep. And in fact, when you take antidepressant, it increases REM latency. When you talk about Alzheimer's, the slow waves during deep sleep and impairment in those waves are been associated with Alzheimer's. So today, if you want sleep stages, you send someone to a sleep lab. This is MGH, and you see they put electrodes on his head and body. He's not happy. It's hard to imagine anyone being happy sleeping like this. So let me show you what we have. So this is our device. It transmits very low power wireless signal, analyzes the reflections using AI, and from that, it gets the hypnogram. So the hypnogram is what you get in the sleep lab. For every 30 seconds of sleep, it tells you the person now is in light sleep, in deep sleep. Now they are moving to REM. Now they are exiting REM. For every 30 seconds, it tells you that. And this is exactly what we get from just the wireless signal and AI. And again, we compare to the gold standard, and in the case of sleep, it's something called PSG with all of those sensors that you saw on the previous slide. So this person is sitting like you guys, and what you see on the screen is nothing but his inhales, exhales. And we ask him to hold his breath. You see the signal now is at a steady level because he exhaled, he did not inhale. And again, for everything that we do, we compare to the gold standard, which is, in the case of breathing, is an FDA-approved breathing belt on the person. And we show that we are highly accurate. So over the last 3 years, we have been working with medical doctors, with pharmaceutical companies, and with different entities in the healthcare domain, and we have been monitoring patients in their home using these devices. So as we are talking now, there are many hundreds of these devices monitoring patients sitting in the home of actual patients in clinical trials for some new therapies as well as some natural history, trying to understand how these diseases are changing patient and the therapies are changing the physiology of Parkinson's, Alzheimer, depression, Crohn's, FSHD, Rett syndrome, atopic dermatitis, lupus, IBD. So as you can see, in fact, we are using this not only for neurological diseases, we are also using them for things that are related to inflammation and immunology to understand the dynamics of the immune system and the response to inflammation. But I want to focus today on neurological diseases. So let me start right now. I showed you results from people in the lab. Let's talk about results from actual patients in clinical trials. And I'm going to show you results starting with Parkinson's disease. So, and they come from these two papers. A paper, one of them in Science Translational Medicine and the other one in Nature Medicine. So the first thing that is very interesting to understand is, can we detect Parkinson's early? Right? So Parkinson, many people don't know that Parkinson is actually the fastest growing neurological disease in the world today. So it is the second most common disease after Alzheimer's. And like other neurodegenerative diseases, Parkinson and Alzheimer, we tend to detect the disease late after the onset in the brain maybe started 10 years ago. Right? And that's really bad because there are many, there is literature, the research literature shows that by the time we detect Parkinson's, the dopaminergic neurons in the brain are 50% to 80% already impaired, okay, already damaged. Okay, so if you are diagnosing a disease when the neurons, the dopaminergic neurons associated with the disease are already 80% damaged, how much chance do you have at really curing or stopping the disease or doing anything useful? Okay, so we really need to detect Parkinson early, but one of the reasons why, and maybe the most important reason why we detect the onset of Parkinson late is because we use the motor symptoms. So people start having tremor, they start having stiffness of motion, and then we detect from that they have Parkinson's. So if we are to detect Parkinson early, we need something different. So we ask a very simple question. Can AI detect Parkinson's from someone breathing, particularly nocturnal breathing, when they go to sleep, how their respiratory signal changes? And in fact, even as early as the work of James Parkinson himself, he reports that the breathing changes in Parkinson's. But of course, when I asked my collaborator, Dr. Ray Dorsey, it's like, oh, can we actually, do you think that it's possible to diagnose from the breathing? He said, good luck. Okay, but can AI detect Parkinson's from nocturnal breathing? So we took respiratory signal. We trained a neural network to answer two questions. Does this person have Parkinson's disease? And if they do, what is the severity of their disease according to the gold standard, which is, in the case of Parkinson's, something called the MDS-UPDRS. This is a large study. We pulled our wireless data as well as data from sleep lab from MGH, from Mayo Clinic, and University of Rochester Medical School. And let me show you the results. So these, I'm sure, at least the vast majority of you guys are very familiar with these curves. They are called the rock curve. On one axis, you have sensitivity. On the other axis, you have specificity, or 1 minus specificity. What is important here is the area under the curve, that number, the AUC. And what this tells you is, with an accuracy, you can interpret it as an accuracy, with an accuracy that is 90% from someone's breathing, taken from wireless signal, we can tell whether they have Parkinson's or not. Now, this study, like actually, as I said, it's a large study that involves 7,000 individuals. 700 of them have Parkinson's disease. So we know that this is actually real, and there are like data that comes from different sites. The other natural question to ask after that is, can AI detect Parkinson's before clinical diagnosis? Because that's really the hope. And of course, this is a harder question to ask, because how do you know, like, if I test someone and the AI system says that they have Parkinson's, I mean, I have to wait for so many years to detect that they actually developed Parkinson's or not. So what we did, we found a public data set that has a respiratory signal from 2 visits. And the first visit is separated from the second one by 6 years. Okay? So what we did is we said, let's apply the AI to the first visit and look at the people who later, in the first visit, they didn't have Parkinson's, but later, 6 years later, they came and said, I was diagnosed with Parkinson's. And let's see whether the algorithm can detect them 6 years earlier, before they got any diagnosis. So here you can see the result. So there are about 500 individuals in this data set, and you can see the people who eventually, 6 years later, they came and said that I have Parkinson's, I was diagnosed with Parkinson's. You see them in blue. And the algorithm, much earlier on the first visit when they had no diagnosis, is saying already, I think those people actually do have Parkinson's, or at least 75% of them are already diagnosed with Parkinson's. While the people that didn't have Parkinson's but didn't develop Parkinson's, you can see the algorithm is pretty sure, no, they don't have Parkinson's. These are the ones in red. So that's great. That's actually give us hope. I mean, of course, we have to build on these results and expand further, and we are actually like building a large collaboration now with MGH, with the Brigham, with Mayo Clinic to like go for like exploring this even further. But this is very encouraging that AI can give us information. It's not about replacing the medical professional. It's really about new opportunities in medicine that we can't reason about today. Okay, so another question is progression in Parkinson's. So if you are a pharma company developing a drug for Parkinson's, and by the way, we don't have any drug that cure Parkinson's or stop the progression of the disease today. There are no drugs for Parkinson's. The only drugs that we have are just drugs that deal with symptoms. But if you want to develop a drug or therapy, you need to be able to differentiate between the progression of the arm that is on therapy and the progression of the arm that is not on therapy. And progression is very slow in Parkinson's. So what you end up having, that you need very large data sets. You need like thousands and thousands of individuals to be monitored for years before you can make any decision. And that naturally brings so many reasons for the clinical trial to fail. And we still don't have any real drug or therapy for Parkinson's. So let me show you a progression biomarker, digital biomarker for Parkinson's. So here, I'm going to start with just like exploratory graphs. So here what you see on the x-axis is time. So this is about 1 year, so 12 months. On the y-axis is the gait of the person, like the speed at which they walk. And I'm just showing an example of a Parkinson patient and someone who's the same age but does not have Parkinson, a control individual. And of course, like this, the person who has Parkinson is slower, but that's not the important part. The important part is that the person who has Parkinson is slowing down, like the gait, like you see the curve goes down twice as fast as the person who doesn't have Parkinson over the same period. So if we are in our 80s, like when people are old, we all slow down. So every year we are slower than the year before. Okay? That's normal. But if you have Parkinson's, then you're going to slow down almost twice as much as somebody your age over the same period. Now, those were like 2 individuals. Maybe there is something special about those individuals. So let's see the cohort. So we did a linear mixed effect model over the cohort, and you see the control in blue and the Parkinson in red. And this is actually the first time that anyone was able to show a statistically meaningful digital biomarker that can differentiate progression of control from Parkinson individual. over this period of time, and only we have 50 individuals. Very small data set. So why is this important? Because as I told you, we really want a therapy. We want a drug or therapy for Parkinson. We want to be able to deal with this disease. But if you are in this business of developing drug for Parkinson, you can put on the x-axis the probability or the percentage or the effect size of your therapy, and on the y-axis, the size of the clinical trial that you need if you want to monitor individual and detect whether they progressed or not over 1 year. And you see, like, if you have a therapy that is 30% effective, actually you are golden. Like any pharma or biotech company will be jumping up and down if they have such a drug or therapy. But to be able to show statistical significance on such a clinical trial, you need about 11,000 participants if you want to get the statistical significance within monitoring of 1 year of the patient. So what happens? Most people just cannot afford having that number of patients, so they run very long, like monitoring people for 5 years and more, and still they need 1,000, but maybe not 11,000. Let me show you what we, like if you use the gait marker, so more than an order of magnitude smaller size of clinical study, and you can say with statistical significance that what I'm seeing here is not just randomness. Is actually statistically reproducible. Okay, we talked about neurodegeneration, but of course, I mean, mood disorders are very important, and there are many results that show that, okay, maybe in many of these antidepressants, what's happening is just a placebo effect. How do we know? If we can't measure the effect of your antidepressant, how do I know that this is just in someone's head? Or, I mean, it is actually in someone's head either way. But how do I know that this is real versus like, okay, so they probably had a fight with their girlfriend and this is why they are extra depressed, or it's not the drug, whether it's effective or not. So we asked this question to a neural network. Can actually we take, again, we are taking a radio signal, we are transforming, we are starting with breathing signal that can come from radio signal or can come from breathing belt, we don't care, but it is nocturnal breathing. We train a neural network to answer a question, is this person taking antidepressant or not? Now, of course, I mean, you can ask the person, I mean, you don't really need this question on its own is not interesting. I mean, you know who's taking antidepressant and who's not, unless they are not compliant. But, and this is another benefit of this. But the real thing that if the machine learning can tell that the person is taking the antidepressant, it's not because it's seeing them putting the drug and taking the pill, it's actually because the physiology has changed. Something about their respiratory response has changed as a result of taking the antidepressant. And so let me show you. So this is, here you see this is a study with 2,000 individuals. 200 of them are taking antidepressant. This is ongoing work as opposed to the previous one that are published. But already you can see from the ROC curve, as I told you, sensitivity, specificity, but the important number is the AUC. So with an accuracy that is 86%, machine learning can look at someone breathing during sleep and say, taking antidepressant, not taking antidepressant. And in fact, we tried a variety of antidepressants, like whether you are using SSRI-based antidepressant or SNRI or others. The vast majority, anything that we tried, or the vast majority that we tried, actually are really responsive to this AI algorithm. So let me show you just an example. So here on the x-axis, you see time. This is an example of a person who was put on Prozac during the monitoring period. So you see, you see time on the x-axis, on the y-axis is the output of the machine learning. And you can see, like in the middle, at some point, the machine learning, the orange curve just goes up and crosses the 50% threshold, saying, okay, so at the beginning, it's saying like, no, this person is not taking antidepressant, not taking antidepressant. But then at some point, very quickly, it rises to cross the 0.5, which is saying, yeah, I think this person is taking antidepressant. But then the other thing that's really interesting to me is that it keeps rising higher and higher over time, which we know that the effect of antidepressant are not just like don't happen only like when the person takes the antidepressant. There is something that builds up over time, like a new synapses that are changing over time. So that actually kind of like is showing in this neural network that it is discovering extra evidence over time. I want to end with talking about generative AI. Now, of course, all of us are very excited about generative AI. I'm sure all of you guys have used ChatGPT. That's really interesting that we can generate such important modality at such accuracy using machine learning. But I want to talk about generative AI in medicine and how we can do it, how we can use it. So today, you can give a prompt and it generates either text or images. So in ChatGPT, I prompt with text, it generates much bigger, more extensive text. And in images, you prompt with text, and it generates an image. For example, I can say, generate for me a corgi that is playing trumpet, and DALL-E will generate that for me. Now, the real question, can I generate clinical data? Can I generate data that have medical importance that reflects the physiology of the person? So I want to give as a prompt the person breathing that I can collect from wireless signal completely passively. So think about breathing like text. And I want to generate their EEG signal. Why is this even possible, or how is this related? I think this is very much related to this image here, because if you think about the text, it's really the caption of the corgi. Okay? So the text and the image are consistent with each other. And similarly, someone's breathing and someone's EEG at the same time have to be consistent with each other. Your EEG cannot be completely in a different place if you are breathing in a particular way, particularly if it's nocturnal breathing. Okay, so let's see some of the results. This is also ongoing work that's very exciting for us. So generative AI for brain EEG. So this person has Alzheimer's. This is actually their actual ground truth EEG spectrogram. And this is what the model, the generative model, generates for them. Now, if you deal with EEG in your practice, you will see that this is highly accurate, but visually, this is very highly accurate. But generating EEG spectrum is not like generating images. Like in an image, like if the AI generates a cat without an ear, we as humans, like me, you, anyone can look at it and say, the ear is missing. But if we generate EEG, if we generate clinical data, particularly if we want to understand from it something about medicine that we don't know yet, even the expert, like I show you about Parkinson and the breathing, then it's not easy to just look at it and say, it looks good to me. I want to know that it has the same power as the original EEG for answering clinical questions. So that was the next thing. What if the generated EEG can detect Alzheimer's better than the ground truth EEG? Do you think this is feasible or this is possible? It turned out actually that it can. So here what you see is the accuracy, which is computed again as AUC. If the ground truth EEG, we went and trained a neural network on the ground truth EEG spectrogram to answer the question, does this person have Alzheimer's or not? And we trained on the generated EEG, and actually the accuracy on the generated EEG is higher. Now, of course, this is really interesting. Why? Why is the accuracy on the generated EEG is higher? And I'm happy to answer this, take that as a question if people are interested in why, what are the potential hypotheses here. But it tells us that this EEG is highly valuable. In fact, I showed you that this model allows us to understand and detect Parkinson, understand and detect Alzheimer's. In fact, we are using it to also detect, like whether somebody is taking antidepressant or not, other types of drugs and therapy. So really, it's what we call foundational model. What we are looking at doing now is basically thinking about physiology as a language. Okay? So similarly to how we have our spoken languages, and like English or Chinese, we have a grammar, we have a language, things are not random. If you have a sentence, you have certain expectation. If what is the next word in a particular sentence and what is the next sentence, there are rules that are related to the grammar, but also rules that are related to semantics. You can think of your physiology similarly, that there are rules in that controls these physiological signals, particularly during sleep when the brain is not actively interfering, like you are basically running the system based on its physiology. So when you're breathing, like our breathing, our EEG, our heartbeats, they have building blocks, similarly to how language has building blocks. There is a grammar that, although it's not spoken, but there are underlying rules about how these physiological signals can change as a function also of your age, your gender, your current health state, your chronic diseases, and your acute conditions. All of those things are not random. They are controlled with certain rules. We just don't know the rules. And not only this, similarly to how there is consistency between an image and its caption, there is also consistency between different physiological signals at the same time from the same individual. Your breathing and heartbeats don't go randomly when you have your EEG in a particular form. So similarly to how I can train a model to understand Chinese, although I don't speak Chinese, I can't read the Chinese character, I don't know anything about the grammar or the semantics of Chinese text, but I can train a model to do that. We believe that we can train models to do this for physiological signals with, of course, we don't know exactly how much power, but we are seeing that these models are able to tell us whether someone has Alzheimer's with fairly high accuracy, whether someone has Parkinson's with high accuracy, the drugs that they are taking, or at least some subset of those drugs. So this is where we are going with my lab, basically building foundational models, but instead of thinking of ChatGPT working on like medical records as the foundational model for medicine, we want to work on foundational model for clinical AI for physiological signals. Thank you. I'll take any questions.
Hi. It was a great talk. I was really impressed. I was wondering, those signals that AI is capturing for like patient, whether they have Parkinson or like taking antidepressant, is that those signals can be seen by human eyes, like doctor's eyes?
Yeah, so I think there are, it's the question, basically, can we see something that is interpretable from the feature that the model is using? So this is one of the things that we are working on. So we believe at this stage that there are interpretable features or feature that you can see visually or pattern that we can show visually that are associated with Parkinson, that are associated with Alzheimer, and that they are compatible with the medical literature. But I want to emphasize that it might not be all of the features. I guess, I mean, the model may have things that cannot be seen by us because it's like something that happens here with something that happens there and something that happens, like it might be very dispersed patterns that it's hard for us to see. But yeah, we have at this stage features that we can show to people.
Yes, so I was really wondering, is that the AI capturing the signals that like people cannot see or not? And also I have a second question. For the predicting Parkinson's 6 years before, did you use the breathing signals?
Yeah. So in all cases, we use breathing signals because, of course, we have limited data set if we are just to rely on the wireless signal. So what we do in AI, which is to create an intermediate representation so that we can take wireless signal and transform to the intermediate representation, or we can take sleep data from the sleep lab, which has typically a respiratory belt, and we can use that to augment the data. So yes.
Thank you very much.
Sure.
So fascinating talk, Dr. Katabi.
Thank you.
So I have a couple of questions, right? So in terms of the wireless sensing, is it affected by obstacles or like multiple people in the room? Because that's sort of the holy grail of sensing, right? If you have something ambient, you don't have to wear anything. And the second part of that question is it complementary to or completely replaces sort of wearable sensors?
Yeah, so basically the vast majority of the people that we monitor have partners and bed partners, and so yeah, we can get this data and these results in the presence of other people in the environment. Now, many people may not know that wireless signal can be made directional. So kind of like you zoom in on the voxel of space that has a participant of interest, and you just limit yourself to wireless signal from that voxel, and you move with that voxel. So that's the answer to your first question. Your second question is whether this replaces the wearables or complement the wearables. I think the second. Basically, the more data, the merrier. I think the wearables, of course, can go outside the home and monitor people on the street and all of that stuff. But I think we call them the emerald technology, kind of like a new class of what we call the invisibles. So I think they have like a role in sense that it is harder for like with the wearables, there is one of the things that happens, which is a natural bias. Like you bias what you are measuring by the fact, like when the person uses the technology, who are the people that are more willing to use that technology. The typical thing that happens when people are sick, they don't have the power to put the device on them, and therefore you get data that is biased in a particular way. Always, not just me, we started with a wearable, and that was one of the things, is just the noise to this, the noise is much higher in the wearables. The signal to the noise ratio is much lower than when you just let people live their lives and you are not differentiating between who's really technology savvy, who's not, what time they are willing to put a wearable on them, when they were able to charge it, all of that stuff. Yes, Joe?
Yes, thank you. So this is a question from our virtual audience. The question reads, hi, I'm Yosra Magdi, based at Qatar University. I build machine learning solutions and I'm a medical student. How do you eventually plan to benchmark and then possibly trial these solutions in hospitals? And I'd go a little bit farther than that, say, at any clinical point of care. Have you been thinking about this?
Yes, so I think this is really important because I think we are at a time when we know that the signal is accurate, the results are accurate, they have been tested in clinical trials, they have been tested against the gold standard. It's very important to collaborate with hospitals, with institutions, with more with medical doctors. We are currently collaborating with medical doctors. But in the hospital, you have many, like just in the hospital, there are applications for inpatients and outpatients. So for inpatients, for example, you're talking about when people go to the ward, can I monitor their physiological signals? Can I understand the relation of sleep to recovery? Can I understand, for example, people who are getting breathing depression as a result of opioid and react to it much earlier than they end up in bad state? Can I detect sepsis or the transition, how the inflammation is starting? And this is, again, as I said, I didn't present anything today about inflammation, but we do have results that are separate that come in the inflammation domain. So yeah, there are many applications in the hospital, and I think naturally that is one way to collaborate. And the second thing is collaborating on connected health. So of course, as when you have a hospital, you want to be able to send the patient home as soon as possible. But now you discharge those patients, you lost any data from them. What if you discharge them with an emerald device that you can keep collecting the information from them at home, and you can keep reacting to it. Also, there is in chronic diseases, like people who have COPD, CHF, can we detect exacerbation early on and prevent it, give the right response? Next.
Yes, I'm Andrew Vickers. I'm a biostatistician at Memorial Sloan Kettering Cancer Center. So that was one of the nicest talks I've ever heard, because I love remote monitoring and I hate wearables. And we've actually been trying to implement them, and it just doesn't work in exactly the sort of ways. I could have said, this is a comment that I'm going to make that I could have made on any talk, but I want to make it to yours because I think yours is one of the most important. I kind of feel my role here is to say, you know, I work in medical prediction, and there's a lot of machine learning people who work, you're doing stuff that I could never do, but I think there's a lot to learn from the medical prediction field. And everybody is presenting sensitivity, specificity, and AUC, and actually in the medical prediction field, people are like, no, that's kind of not very important, right? The standard way we look at it is discrimination, calibration, clinical utility. And I would just encourage, if you're going to develop these, you have to look at calibration and you have to look at clinical utility. And there's very easy stats that you can present to do those. So it's just a way of kind of trying to bring this into the mainstream of medical prediction.
Yeah, no, that's actually a very interesting question, and thank you very much for asking it. So in fact, yeah, I do work on in 3 communities to some extent, like the medical doctor, the biostatisticians, at least 3 communities. I work with others, but that are relevant here, and the AI and machine learning people. And in fact, maybe you have noticed, I talked about when we were talking about clinical trials, I talked about mixed linear effect models. Like basically what you want to see, you want to like be able to take your biomarkers, first do like say, okay, what is the context of use and what is the actual biomarker, and then use like standard ways that we have like developed a whole like theory and literature and like so much about how we can use statistics, not just to say that it works in this population, but basically be able to understand how it's going to perform in a new population, that it's not just what I'm seeing here, that this is meaningful statistically, and there are many models depending on the relationship that you are trying to achieve. So yes, I tailor a bit the talk to the different audience, but I do think that there is a lot to be done by combining the statistical analysis with the machine learning power to extract new biomarkers, but you put on top of them statistical analysis to be able to understand where and what is the exact power of these models.
Hi, Alex Lupi from France. I'm a physician and I'm also a biostatistician, so some of my questions were already addressed. So I have a question about your Parkinson prediction model. I like it. I think it's important. So how do you comment on the potential selection bias that you may have, because you selected 700 Parkinson patients out of 7,000, which is not the distribution and the prevalence that the general population will observe. So there might be some kind of selection bias. And my second question is also related to the performance metrics. The AUC that you find might be inflated by, you know, the overperformance of your classifier. So do you have any plans to show the generalizability and try to actually put the performance metrics in some kind of external validation course? I think that will be reinforcing your results.
Yeah, so I think there are a bit of different things in this question. So one thing is like when you're training a model, independent of whether like in Parkinson, for example, the typical percentage that people believe in the population of older individual is about 2%, something like that. And so one thing is when you do the training is like if you use 2% versus much larger control, in fact, that is not good training for the machine learning, because during training, your interest is not to balance the same thing that you see in the population. Your interest is to learn what are the features of each one of these two populations. So as much as possible, you want to like balance the two, but at the same time, use all the data that you have, because any data that you have can bring new information.